Welcome to our channel, Mootechie. This series begins with an overview of numerical taxonomy, a fascinating quantitative approach to classifying living organisms. Moving to the next topic, let's define what this field entails. Introduction. What is numerical taxonomy? A biological classification system based on overall phenetic similarity among organisms rather than evolutionary history. This means it groups organisms by how similar they look or function right now, not by their shared ancestry. It analyzes operational taxonomic units or OTUs using large sets of observed characteristics. An OTU could be an individual, a species, or any group we want to classify. It utilizes mathematical algorithms to compute similarity coefficients, which are numbers that quantify resemblance. The final result is a phenogram, which is a tree diagram that groups organisms into clusters. This approach is also known as Phenetics, pioneered by Sokal and Sneath. Figure 1 shows the conceptual phenogram. We see clusters like OTU-A and OTU-B grouped together, and OTU-C and OTU-D forming another cluster, visually representing their calculated similarity. Now that we know what it is, let's explore how it's done. Methodology. More principles. Equal weighting. All characters are considered equally important a priori to eliminate subjective bias in selection. This principle prevents a researcher from unconsciously giving more importance to one trait over another at the start. Maximal data. Utilizes a large number of independent characters, often more than 100 for statistical robustness. The more traits we compare, the more reliable and stable the resulting classification tends to be. Phenetic similarity. Grouping is based solely on observed overall similarities, ignoring evolutionary ancestry. It focuses on what we can measure today, not on inferred historical relationships. Reproducibility. Explicit mathematical algorithms ensure different taxonomists reach the same classification. If you follow the same steps with the same data, you should get the same result, which is a key strength of science. Synthesis. Combines data from multiple sources like morphology, physiology, and chemistry into a unified matrix. This holistic view allows for a comprehensive comparison across different aspects of an organism. The graphic shows a cycle, from collected data through numerical methods and algorithms leading to an observed result. To understand this methodology in action, let's walk through the standard process. Process flow. Steps in numerical taxonomy. Step 1. Select OTUs and select characters. Choose the organisms and the specific traits you will compare across all of them. Step 2. Code data. Convert traits into binary or multi-state codes like present as 1 and absent as 0 so a computer can process them. Step 3. Compute a similarity matrix. Use a formula to calculate a score for how similar every pair of OTUs is based on all the coded traits. Step 4. Cluster and build phenogram. Apply an algorithm to the similarity matrix to create the branching tree diagram that shows the groups. Step 5. Validate. Test the stability of the clusters and refine the results, perhaps by using different algorithms or checking for errors. After seeing the process, let's consider why this approach is so valuable. Benefits and merits. Advantages of numerical taxonomy. Greater objectivity. Explicit numerical methods minimize subjective bias in classification compared to traditional methods. It reduces the influence of personal judgment, making taxonomy more scientific. Large data sets. Can handle huge amounts of data, integrating morphology, anatomy, and chemistry into a unified system. This allows for complex analyses that wouldn't be possible by eye or intuition alone. Reproducibility. Classifications are statistically testable and can be repeated by different taxonomists with the same results. This consistency is a cornerstone of good scientific practice. Better keys. Provides clearer group limits and better identification keys for biological taxa. This directly helps field biologists and students identify unknown organisms more accurately. Automation ready. The data format is perfectly suited for computer automation and rapid analysis of new specimens. This makes it highly efficient and scalable for modern biodiversity studies. Of course, no method is perfect. It's important to understand its limitations as well. Critical analysis. Limitations of numerical taxonomy. Primarily phenetic. Classifications based on similarity may not reflect true phylogenetic or evolutionary history. Two organisms can look similar due to convergent evolution, not because they are closely related. Subjective character selection. The choice of traits and potential improper weighting can significantly skew results. Even with equal weighting at the start, deciding which traits to include is still a human decision. Data sensitivity. Results are vulnerable to distortion from convergent evolution, also called homoplasy, missing data, or noise in the measurements. These factors can create false signals of similarity or difference. Algorithmic dependence. Different clustering methods, for example UPGMA versus single linkage, often yield deflecting phenograms. The choice of math can change the picture, which requires careful interpretation. Quantity versus quality. Risk of prioritizing the sheer number of traits over their biological significance or stability. 100 trivial traits might not be as informative as 10 fundamental reliable ones. Finally, we wrap up our overview. Thank you. We hope this helps with your biology exam preparation. For more visual lessons, Mootechie. Subscribe, like, and share. Keep learning, keep exploring.